Hello, everyone, and welcome to the podcast, A Journey Through Nature and Science. I'm your host, Don Murphy. This episode is all about the technology that has revolutionized everything from ancestry research to tools that have improved the criminal justice system and helped to set many innocent people free. Known as PCR, or the polymerase chain reaction, Dr. Henry Erlich is here to tell us how he and his colleagues discovered PCR and how it is used today. Dr. Erlich is a world-renowned scientist, author, educator, and human rights activist. His contributions to the field of DNA research are really groundbreaking. In the late 1980s, he pioneered innovative techniques for assessing DNA evidence, revolutionizing the criminal justice system. Under his leadership, he and his colleagues developed the polymerase chain reaction, or PCR technology. The technology is used in numerous ways and is familiar to people even in their daily lives. PCR technology has helped save millions of lives worldwide. But beyond his work in DCA forensics, Dr. Erlich's research has contributed to medical research and enriched our understanding of evolution and historical events. His latest book is DNA Analysis and Human Evolution, Genetic Reconstruction of the Past. So, Dr. Erlich, welcome to the program, and it's great to have you here.
Well, thanks a lot, Don. It's a pleasure to be here with you.
Yeah, appreciate it. So, what started you on your personal journey? What led you to work in molecular biology and biochemistry? And was there something that happened in your youth or childhood where you said, hey, this is the direction I want to go in? And then maybe also tell us what got you started specifically with PCR technology.
Well, in terms of biology and molecular biology, it was really a course I took my freshman year in college. That in high school, I actually never even took a biology course, but I'd always been interested in it and read about it. I took physics and chemistry, but never biology. But in my freshman year, I was actually majoring in Renaissance history and literature. And I took an introductory course in biology that was taught by a really brilliant scientist and a charismatic teacher. His name was George Wald, and he later got the Nobel Prize for his work on the biochemistry of vision. But he was mainly a really inspiring teacher, and it basically inspired me to switch my major in college from history and literature to biochemistry. And then I was very lucky to take a genetics course from another brilliant scientist, Matt Messelson. So I ended up, so that was a decision I made, not so much as a career choice, but just because I found the area fascinating. But of course, it turned into a career choice because I went on to get a PhD in genetics later.
Right. You mentioned Messelson. I mean, he's pretty famous in his own right. He did some really important work and experiments early on in genetics.
Yes. Yeah, no, it's good that you point that out, Don, because probably the most beautiful, elegantly designed experiment in molecular biology that introductory biology students all learn is the so-called Messelson-Stahl experiment, where the experiment was designed to investigate how the DNA replicates. So as you know, DNA is a double helix. So there are two strands, but they showed that the way DNA replicates within the cell is that the two strands become separated, and then each of the separated strands serves as a template for the synthesis of a new strand. So that, and it was a really beautiful experiment in the way it was designed and the controls. So it's often taught in biology classes as an example of experimental design.
Yeah, I remember very well in my days. And when you say strand of DNA, it's really important, I think, for our listeners to understand. When we're talking about a strand, we're talking about a series of what are called bases or biochemical molecules. And people often see A, T, C, and G. And if you wonder what those things mean, they're letters that represent biomolecules that are strung together on these strands, and the three of them together code for a particular gene. Is that right?
Well, yeah, so that the four bases A, C, T, and G are strung together on a strand, and the what Watson and Crick showed with the double helical structure of DNA was that they're two strands, and they're bound by what are called hydrogen bonds. So the interaction between an A on one strand and a T on the other, and a G on one strand and a C on the other, those are what's called base pairs. So that's what holds the double helix together. And then, Don, as you mentioned, the genetic code basically is a way of translating the sequence of bases into the DNA into the synthesis of a protein. And a protein is made up of amino acids, and the basic genetic code is that three bases, let's say ATG, that would code for an individual amino acid. So the genetic code that's in the DNA is instructions for how to synthesize a protein. And the way that's done is that another biomolecule called RNA or messenger RNA is transcribed or synthesized from the DNA strand, and then that is what's called translated into a protein. So the information in the DNA results in the synthesis of a protein through this intermediary molecule known as messenger RNA.
Right. Absolutely. Well, this is critical for understanding then how the code or the actual gene that you look at or genetic markers are actually these chemical bases strung together, which is just absolutely fascinating. And then you and your colleagues did work to discover how to, I guess, make a whole bunch of copies of these individual genes. And how did you get involved in that? I heard a lecture that you gave once that had to do with a bus ride from somewhere back to California to start working in a lab on this in either as a postdoc or a young doctorate student. How did you get involved in specifically with PCR?
Well, after I graduated and got my PhD in genetics, I did one postdoc at Princeton looking at bacterial replication. You know how bacteria replicated their DNA. You know what factors were involved in generating mutations and so forth. But I took a course when I was at Princeton in immunology, and I become fascinated, with the complexity and mystery of immunology, and also the possibility that if you studied immunology, you could make real contributions to medicine, because of course immunology was intimately related to disease and health. So I did a second postdoc in immunogenetics at Stanford, and then and the part about the bus in California might have to do with why I took a job at a little biotech company. So I had moved to California, and I was doing a postdoc at Stanford, and my girlfriend at the time was a professor at Oberlin College in Ohio, and
Yes.
so she had a dance company, and she moved to California to be with me in a big yellow school bus with 11 dancers and her three kids.
Oh my goodness.
We now have four, so I made a genetic contribution myself there. Um, but anyway, she then came out and established a dance company, which is now a world famous school and dance company and theater in San Francisco. But because she moved out to be with me when I finished my postdoc at Stanford, I basically had to take or find a job in the Bay Area. And there were these new biotech companies that were just starting. Genentech was a tiny little startup at the time, and Cetus was another little tiny biotech company. So I took a job at Cetus. And as I write in the preface of my book, I was worried about going to a corporate entity, a company, but the culture at Cetus turned out to be a lot more counterculture than corporate culture. So
That's
we
great.
were kind of a bunch of hippies trying to figure out what to do. And one of my colleagues, Kerry Mullis, had this idea that he called the polymerase chain reaction, or PCR. So we started working together, and Kerry had this idea of how it should work, and he never actually was able to demonstrate that the idea worked. But I was working in parallel with Kerry,
Mhm.
and we were able to show my colleagues and I that you could amplify the beta globin gene from human genomic DNA. And we decided
And when
as
you
a
say amplify, you're meaning make a bunch more copies of it, right?
yeah, no, you're right, Don. Thanks for the clarification. Um, so we initially started, I was trying to develop a DNA technology that could be used for prenatal diagnosis in sickle cell anemia and beta thalassemia. And those are both diseases that results from mutations in the gene for beta globin, which is a protein that's involved in hemoglobin and the transport of oxygen
Right.
within the body. So we decided that our test for this idea that Carrie had, PCR, we decided we could see if we were able to use that idea to amplify or to synthesize millions and millions of copies of a specific DNA region. And we picked the beta globin region because we wanted to carry out this diagnostic test for sickle cell anemia and beta thalassemia.
Right.
And the idea, I'm glad we discussed the Mendelssohn-Stahl experiment, because the idea of PCR really uses the strategy that the cell uses to replicate DNA.
Hmm.
So that the idea of PCR is you start out, of course, with a double-stranded molecule, and then by heating the DNA, you can separate the two strands. So you
Hmm.
now have two separated strands. And then using an enzyme known as DNA polymerase, which is what's used within the cell to replicate DNA, you can use that enzyme to synthesize a copy of one strand and to synthesize a copy of the other strand. And so that's what we call one cycle of PCR. In one cycle of PCR, you've doubled the amount of DNA that you started with. And
Right.
so this is basically an exponential reaction. You keep doing cycles of PCR. So after 20 cycles, you synthesize in a test tube 1 million copies of the target sequence that you started out to amplify.
Right.
So that's what PCR is. It's really, as you pointed out earlier, Don, just a way to synthesize millions and millions of copies of something so that you can then study it. And
Right.
so, for instance, in our forensic application, because we were using PCR, if there was a tiny blood stain or a tiny shed hair at the scene of the crime, we were able to amplify enough DNA so that we could compare the genetic profile of the evidence with the suspect. So, it's really just a way of making lots of copies so that you can analyze what you've amplified.
Well, let's talk about that for a minute because I think people are really interested. They watch all these TV and detective shows and they talk about DNA amplification and forensics and matching a particular blood stain from a crime scene with that of either a victim or a suspect.
Right.
So 99% of our genetics, our genes are the same. So how do you differentiate between when you're a suspect and you have your blood left at a scene and you're going to look at the DNA on that blood, what are you specifically looking at, and how do you differentiate that between all the other people in the human population that have similar DNA? What makes the difference?
Well, that's a very fundamental point, Don, that as you pointed out, over 99% of the human genome is identical in everybody on this planet. So if you want to use DNA for individual identification, you have to focus on that less than 1% of the DNA that's variable, or the genetic term is polymorphic. That is, there different variants of that gene. So for instance, as I said, when we were developing PCR in the mid 80s, the very first gene we ever amplified was beta globin for diagnosis of sickle cell and beta thalassemia. But the second gene that we amplified was a gene that's involved in the immune system. It's one of the so-called HLA genes. These are the genes that if you're having a bone marrow transplant or a kidney transplant, you need to match the HLA genes of the donor and the recipient. And I picked this gene to work on because I was very interested in the immunology system, what governs an immune response. But because this gene was highly polymorphic, that is, had many different variants, or as we call them, alleles in the human population, it could be used for individual identification. And so the way we actually ended up working on the first DNA case in the world in 1986 was in the building where I was doing basic research and trying to apply this new technology, PCR, to medical genetics diagnoses. There was a very small forensics lab in the same building.
Mm-hmm.
And I used to chat with the head of the lab, a friend named Ed Blake. And at one point, he came up to me and said, Henry, I'm having a problem with a particular case. The evidence sample that they want me to analyze has been stored in formaldehyde. So all the proteins are denatured, and I can't use any of the standard forensic techniques that we've been using for years, like ABO blood typing or analyzing the structure of proteins. Those were the standard tests used for identifying samples,
Mm-hmm.
evidence samples in those days. So I said, well,
Understand.
we could try DNA. And so we extracted DNA from this autopsy sample that had been stored in formaldehyde, but the DNA was broken up into little bits.
Mm-hmm.
The term is that the DNA was degraded. Instead of large DNA fragments, you had these little tiny fragments. And so none of the conventional DNA technologies at the time could be used to analyze this particular evidence sample. So I told Ed, well, we could try this new technique that we're developing, PCR, and see if we can amplify this polymorphic HLA gene and see if we could use that to identify the sample. And so the first time we tried it with the DNA polymerase, that is an enzyme from the lab bacterium E. coli.
Right.
And as it turned out, it didn't work. We weren't able to amplify from these small fragments. So I then told Ed that, hey, we're just starting to look at a new DNA polymerase that my colleague David Gelfand had isolated from a bacterium, Thermus aquaticus, that lives in the Yellowstone hot springs.
Right.
So it's a bacterium that can live under very elevated temperatures. And so the enzyme that you get from this bacterium is stable to high temperatures, which turned out to be very important. So as it turned out, that solved the problem, and we could amplify this polymorphic gene and do the identification. So that's how we started out applying PCR to forensics. It was this
Well,
one case.
you're saying a couple of things that are really important, and I'll admit I'm a nerd when it comes to these things because,
Uh-huh.
of course, that's what I studied when I was in college 50 years ago. God, am I dating myself? But nevertheless, you mentioned a couple of things that maybe we can delve into a little bit. You say this HLA gene is polymorphic. So within that one gene, there are different variations of that gene, which must mean that the protein that it ends up producing that's involved with um, hemoglobin and the carrying of oxygen in the body must vary amongst individuals, and
Okay.
the structure of it must be slightly different, but functionally it's the same. Is that right?
Okay, so let me clarify something. So the very first thing we ever amplified was beta globin.
Mm-hmm.
And beta globin isn't really so much polymorphic. I mean, there are lots of known mutations in it, but if you don't have a mutation in the beta globin gene, the sequence is basically the same. So,
Uh-huh.
but the second gene that we amplified was an HLA gene, and that's polymorphic. And you're right that for the HLA genes, the protein that's encoded by these different variants has a different structure.
Mm-hmm.
And the reason that the HLA genes are so polymorphic is basically natural selection. Natural selection has created a diversity of HLA variants so that the human population can respond to a lot of different infectious disease pathogens.
Ah,
Because
nice. Makes sense.
the, the HLA genes encode proteins that basically initiate the immune response. They will bind, for example, a part of a virus and present it to the human immune system to make an immune response and to basically counter that infectious disease pathogen. So that's really why the HLA genes are so polymorphic, because they've evolved to help us respond to a diversity of pathogens in our environment.
All right. That's awesome. And I think that's such a great explanation because I think people wonder why would that there be such variation, and it has all to do with natural selection, that especially with respect to the immune system. That's
Right.
just pretty
Yeah.
awesome. Well, in a very practical sense, what was the first case that you were involved in that set someone free, for example, using this? Because
Okay.
I know
Yeah.
you're very involved in human rights and in criminal justice.
Right. Yeah. As you noted, I'm on the board of the Human Rights Center at Berkeley, and I did it before genetic reconstruction of the past. I did a book with Eric Stover, the director of the Human Rights Center at Berkeley, that was geared more for graduate students and lawyers and judges. So it was pretty technical, both scientifically and legally. But genetic reconstruction of the past, the new book, is for a general audience, and I try to explain, how science works and the assumptions that we make and so forth. But so the first case that was a DNA exoneration was the Gary Dotson case, where Gary Dotson had been convicted and in prison for about 10 years for a rape. And we eventually got the semen stain, the 11-year-old semen stain from the state of Illinois, and were able to show, actually amplifying this and analyzing this HLA gene, that the genetic profile of the semen stain did not match Gary Dotson. And this was about 1988 or 1989. So that was the first DNA exoneration. But as you may know, Don, that since then there have been over 500, probably way over 500 DNA exonerations that have been carried out largely by the Innocence Project.
Right,
But the Innocence Project is a national program started by Peter Neufeld and Barry Scheck.
right.
But there are lots of local or state innocence projects. So there have been a lot of DNA exonerations, and in I think over 25 or 30 of the cases, the people who've been exonerated were on death row.
Mm,
And actually, I myself was involved in a very dramatic case in, I think it was 1994, where Earl Washington Jr. in the state of Virginia had been convicted of rape and murder and was scheduled to be executed. But I could show that the DNA actually excluded him, so he was exonerated.
Hmm.
Um, but that was a long process. I mean, one of the things I learned in the Earl Washington Jr. case was that sometimes confessions are coerced. So Earl Washington Jr., who was cognitively impaired, had confessed to this crime, but at the trial, he said no, he was innocent. And so that was an example of a coerced confession. And in
Sure.
several of the cases that I've been involved in, there was a false confession. So that was a surprise to me. I naively, well, I was unaware that the incidence of false confessions was so high.
Right. Now you mentioned that in this case, or was it another case, or this case with Earl Washington Jr., where it was an exclusion, his DNA was excluded because there wasn't a match. But sometimes there's what's called inclusion, and that's a more difficult situation.
Well, right. So Don, when you compare the genetic profile of the evidence with either a victim or a suspect, the result is either a match or an inclusion, or they're different, and you can say it's an exclusion. If it's an exclusion, the statistics are very easy. You can say, no, this blood stain did not come from this suspect. So you don't even need statistics. But if they match and it's an inclusion, you have to make some calculation. Basically, what is the likely of a coincidental match? That is, what is the
Right.
likelihood that someone else, not the suspect, would also match? And the technical phrase or term for that is the random match probability.
Hmm.
And what that means is, what is the probability that if you picked some individual from random, at random, from a specified population, that he or she would also match?
Right.
And that's crucial. And, before DNA, if you got a blood type match or looking at the protein structure match, that might be, 1 in 100. There might be 1 in 100 chance that someone else would also have that. So, that's helpful. I mean, that's meaningful information, but certainly it's not what a lawyer would call dispositive. It's not deterrent, it's not proof that that sample came from the suspect. But, with DNA, when you look at lots of different markers around the genome, uh, and you can multiply the probability of a match at each marker, you can generate numbers like 1 in 100 million or 1 in 1 billion. So that's pretty convincing evidence that the sample did in fact come from the suspect or the victim.
Right. And that's really important in making a difference. There's a variation on this that's going on too that's somewhat controversial, and I know you've talked about this in your book and discussed it in some of your lectures, and that's when you're not just looking at a suspect, but you're actually branching off. A crime has been committed. You don't have a suspect. It may be a cold case or something, and you're trying to find some sort of genetic information. So then you go look at a family tree and you use DNA and you go to a database. And I guess there are a couple of different kinds of databases, but there are ancestry databases out there that you can go look at and see if you get some sort of familia match, and then maybe contact that family member or say that suspect possibly is related to somebody, and that gives you a clue as to who the suspect might be. But I understand that can be somewhat controversial. What's been your experience with that?
Well, basically, database searching started out uh, with, as you say, cold cases or cases in which there is no suspect. So you have an evidence profile, a genetic profile from the evidence, but you have no suspect to compare it to. So the very first cases where this was done was, probably over 20 years ago, uh, because the federal government had established criminal databases. So they started out by having genetic profiles of individuals who'd been convicted of violent crimes. And that was the criminal database. And you could search that to see if you got a match.
Mm-hmm.
Then, and a number of cases were solved that way. So that was a very powerful method of solving suspectless crimes.
Right.
Now, then there was a move that if you didn't find a full match, to do what was called familial searching to see if you got a partial match. And there was one famous case in California called uh, the Grim Sleeper.
Mm-hmm.
It was a case, multiple rapes and murders. And there was a partial match with somebody in the California criminal database. And it looked as if the evidence came from possibly the father of this person who was in the database. His name was Lonnie Franklin Jr. And they went back and they got a sample. Actually, this was an interesting case where they went back and found a discarded pizza box and were able to get
Wow.
DNA from the saliva on the pizza box, and it turned out to be a complete match with the evidence sample. So that was one of the first examples of familial searching, but that was a criminal database. And then, Don, what you were referring to is what's sometimes known as genetic genealogy, where you have a profile from the evidence from the crime scene, and there's no suspect, doesn't match anyone in a criminal database. So you
Mm-hmm.
can then look for a partial match in a genealogy database. And then if you get a partial match in the genealogy database, you can go back and build up a family tree and go back maybe several generations and look at all the people in this family who are related to the person in the database that showed the partial match. And that can be an investigative lead. Then you can look to see at all the people in the family who had the opportunity and means to commit the crime.
Mm-hmm.
And then you can get DNA from them and see if that's a perfect match or complete match with the evidence sample. So that's what's known as investigative genetic genealogy. And it involves the work not just of molecular geneticists, but also genealogists, people who are good at looking at reconstructing family trees.
Right.
And they're probably the most famous case where genetic genealogy solved a really heinous crime that had been a cold case for decades. That was the Golden State Killer,
Mm-hmm.
and that was someone who carried out rapes and murders in both northern and southern California. Um, and it turned out to be a former police officer known as Joseph D'Angelo.
Right. Remember, well, for our listeners, we just actually had a genealogist on the
Yep.
program prior to you, and she talked a lot about the ancestry research and how that's done, and also the controversy where sometimes family members are asked about a potential subject. You know, they get a family member, they're not a suspect, but it gives them a lead, and a family member may feel uncomfortable in terms of giving any additional information that might actually lead to a suspect, and you have to get their cooperation. What's your experience been there? I mean, do families cooperate, or does it become difficult to get information?
Well, I guess the first step is to make sure that the database that you're searching, say if it's a genealogy database, that the people who contributed their genetic profiles have given informed consent
Hmm.
that the profiles can be searched for law enforcement purposes.
Right.
So I think that's a recent development where I think that's now required. You know, actually in the Golden State Killer, they searched a database, and I don't think the people in the database had given explicit consent that it was
Hmm.
okay to search their profiles. But now I think the standard practice is that you can only search profiles for which informed consent has been given. So that's the first step. But there have been critiques of familial searching. One of the critics said that now you're just turning relatives of people in the database into suspects. And I discussed that in the book a little bit. And my feeling is you're not really turning them into suspects. They're potential suspects, but they become suspects only if you follow up the familial searching with a lot of other investigation.
Right, absolutely. Well, switching reels a little bit, you're also doing some important research using PCR in medicine. Of course, you already mentioned that that's kind of how you got involved in the first
Yeah,
place,
right.
is with looking at sickle cell anemia, and now, of course, there's CRISPR, which is being used with the help. But CRISPR wouldn't really be possible without the PCR technology. Isn't that correct?
Well, I think CRISPR is a huge innovation, and it's a real revolutionary game changer, as you know, Don. But a lot of what's currently done in molecular biology sort of builds on PCR or uses PCR as part of what it does. So, for instance, one of the most dramatic things in molecular biology research has been a new, or relatively new, probably the last 15 years or so, 20 years, uh, of DNA sequencing called next generation sequencing.
Mhm.
So it's enormously powerful way of analyzing the sequence in the DNA. And just to give you some examples, maybe 15 or 20 years ago, it took millions of dollars and months and months to sequence a human genome. But
Mhm.
now it can be done for thousands of dollars and, in a week or so. So uh, it's a real game changer. And, it's its own revolutionary innovation, but it utilizes PCR in some of the steps. So PCR has just become kind of an essential tool in a lot of molecular biology technologies, and as you know, it's the basis for a lot of diagnostic tests. So during the pandemic, we were all getting our noses swabbed, and
Right,
having COVID PCR tests.
right, exactly. And one of your colleagues worked on that and helped develop some of that testing. Is that right?
Well, using PCR to quantify the amount of a virus is a very important aspect of the medical applications of PCR. And there are two elements in that. One is, how do you make PCR into a quantitative test, not just qualitative? Is the virus present or not? But how much of the virus is present? And that was the contribution of my friend and colleague Russ Higuchi, who
Mhm.
developed a quantitative method of a quantitative variant of PCR. And then the actual first viral load test was the HIV viral load test, which was created by my other colleague at CETUS, John Snitsky.
Mhm.
And the
Right.
HIV viral load test was really crucial, not just because it could diagnose the presence and the quantity of virus that was present in some individual, but it made it possible to test for antiviral drugs. So if you were testing AZT or any other of the early anti-HIV drugs, instead of just looking at the symptoms of the patient, you could measure the viral load. And if the
Mm.
viral load went down, you knew that the drug was effective.
Understand.
And of course, as I said, in the COVID, there's a quantitative COVID viral load test. And as you may also know that there's a strategy known as environmental PCR, where you look for some virus like COVID in the wastewater. So you
Mm.
can monitor uh, the degree to which COVID is infected a community, not by looking at individuals, but you can look at wastewater and look at the rise and fall of COVID in the environment as a measure of what's happening with the pandemic.
That's just amazing. I mean, that um, I think what's more amazing than anything else, and I hope what people are taking away is how powerful science can be in developing tools that are beneficial to society as a whole. We just discussed a range of very positive things, from potential diseases being prevented or detected in human beings, to understanding genetics well enough to free innocent people from jail, and to prevent mistakes in the criminal justice system, to the kind of monitoring that you just described in communities, to give us information and data so that we can make the right kind of political decisions as well. So
Mhm.
hopefully all of this would lead to making better decisions and understanding. And all of this is the result of science developing these powerful tools. And then as you go back, because I started this talking about what got you involved in the first
Mhm.
place, it has to do with human beings, individuals being
Mhm.
curious and
Yes, yes.
interested enough to follow through on these things and to do the difficult science in the laboratory, driven by personal curiosity. And that's how science is done.
Yeah, no, I agree, Don. I think curiosity is, of course, a major driver of scientific discovery. And I was very lucky to be able to take that course as a freshman because it
Mhm.
changed the course of my life. Uh, and it was really the enthusiasm for discovery and for understanding the nature of the world that George Wald conveyed to his students.
Right. Well, not only did it change your life, but look, I mean, I know you're being modest, but it changed history. It changed the scientific history. It changed the course of history in society, because without these tools, there, as we just described, there's certain things that couldn't have been done, and there are people that would have been affected,
Yeah.
people that may have lost their lives if this technology had not been developed, and of course, not just this technology, but other scientific technology as well, and that's why it's important to do science. There's one other aspect of your PCR work that's being done, and that has to do with evolution. Can you say a few words about what you're doing in evolution, and how, DNA studies and using DNA polymerase in these PCR tests has led now to an understanding of what's happened in the past and how we've evolved as human beings, and not only us, but animals and plants. And it changes our understanding of genetics as well.
Well, my interest in evolution came really when a very bright postdoc came to join my lab. His name was Ulf Ullenston, and we're still friends. He's a professor at Uppsala University now in Sweden, and he came from Alan Wilson's lab. And actually, in my book, I have a whole chapter about Alan Wilson, molecular evolution, and the out of Africa hypothesis, because it's Alan Wilson's lab that studied mitochondrial DNA, which
Mhm.
is mitochondrial DNA is the DNA that's in the cell organelle known as the mitochondrion. And the mitochondrion within the cell, it's in the cytoplasm of the cell, and it generates energy. It's known as the sort of energy factory of the cell.
Right.
But from a genetic point of view, it has its own genome. So it has its own very small uh, 16,000 base pair genome, but it can be analyzed. And what the people in the Wilson lab did was look at mitochondrial DNA sequences from all around the world, and they realized that there was more genetic diversity within Africa than outside Africa. And the pattern of genetic diversity that they documented suggested that, about 75,000 years ago, a group of modern humans emerged from Africa into the Middle East, Europe, and then colonized the rest of the world. And this is all based on the analysis of mitochondrial DNA. And
I
that,
might interject here before you go on is that, and the reason this is possible where you can draw this direct line is because mitochondria DNA comes from the mother or from the female. And as a result, you can trace back from mother to mother to mother in a straight line. Is that correct?
that's right. It's maternally inherited, and it's used not just in evolution, but also in forensics, because a cell has only two copies of the nuclear genome, one from mom, one from dad, but it has thousands of copies of a mitochondrial genome. So there are lots of samples where the only thing you can analyze is the mitochondrial DNA because there's so much more of it. But as you point out, it is passed through the maternal lineage. And so the phrase from Alan Wilson's work, the phrase emerged mitochondrial Eve or African Eve, because all of the mitochondrial sequences, if you made a tree, went back to this one sequence. But the important thing, the other thing that Alan Wilson and some of his colleagues developed was what's known as the molecular clock.
Mm-hmm.
You can compare sequences from, let's say, two different species of the same gene, and if you made certain assumptions about how rapidly things changed, sequences changed, you could infer how far back in time these two species diverged. So you could do that from chimpanzees and humans and estimate that they diverged about 5 million years ago. And then if you compare chimpanzees and humans and gorillas, infer that chimpanzees and humans in one lineage diverged from gorillas about 7 million years ago. And that was all based on the analysis of DNA sequences. And the molecular clock also makes certain assumptions that these sequences are not subject to natural selection, so that they have to be what's called neutral. And one of the chapters in the book discusses how my colleagues and I were in a dispute with another group about the origin of the species. And basically what I argue in the book is that we did it correctly. We looked at sequences that were not subject to natural selection, and they were looking at sequences that were subject to natural selection, which is inappropriate if you want to apply a molecular clock.
But how is how let me interrupt you there and ask how is a particular genome or genetic material not subject to natural selection? How do you make draw that conclusion?
Okay. Well, you can actually look at the sequences and look at evidence. There are different kinds of natural selection. One is what's called positive selection, so it increases the frequency of a particular variant. One is called diversifying selection, which we discussed for the HLA genes, that the selection is to diversify variation at that gene as much as possible. And then one is what's called purifying selection, that all the variants are deleterious. And so if you have purifying selection, that sequence will not change over a long period of time. And there are sort of statistical techniques that you can look at a sequence and make inferences about whether it's subject to natural selection or not. And there is a whole theory and a mathematical modeling of what's called neutral evolution, where DNA sequences evolve or change over time, but just due to the mutation rate and chance, random chance, or
and not pressure, and not some sort of pressure.
That's right. That's right.
I understand. That's
But anyway, one of the things that came out of the Wilson lab was an interest in using PCR for ancient DNA.
mhm,
That since PCR can amplify little bits of DNA from an old forensic sample, Alan Wilson thought, well, maybe we can use this for extinct species or for, fossil remains. And, 20 or 30 or 40 years later, we now have sequenced the Neanderthal genome. We've identified a totally new uh, species, the Denisovans, based on DNA sequence. Now, are you familiar with the story of the Denisovans?
not totally, but I've heard of it.
Yeah. So, as you know, when the Neanderthal genome was sequenced, and of course, as I was saying, everything starts with the mitochondrial DNA because that's the easiest. So, when the mitochondrial DNA of a Neanderthal fossil was sequenced, it was clear that it wasn't human,
Hmm.
but it was very close to human. So you could say, oh, I've now identified the mitochondrial sequence for a different archaic species, Neanderthals. And then Svante Pabo, who actually was a postdoc with Alan Wilson when I was at CETUS and we transferred PCR to the Alan Wilson lab. Svante just got the Nobel Prize a year or two ago for his work sequencing the Neanderthal genome.
Right.
But anyway, as I was saying, there's a cave in Denisova, Siberia, and it had some Neanderthal fossil remains, but it also had a tiny finger bone from which DNA could be extracted. And so when the DNA that was extracted from that finger bone was sequenced, it turned out to be neither modern human nor Neanderthal. And
Mhm.
so it defined an entirely new archaic human species that's now known as the Denisovans.
Mm.
And you can now um, find you may know this, Don, the presence of Neanderthal genome makes up about 2 or
Right.
3% of all human genomes, and
Yes.
Denisovan DNA makes up about 2 or 3 or 4% of human genomes in Southeast Asia.
Right.
So these archaic species interbred with the ancestors of modern humans. So in our genomes, we have both some Neanderthal DNA and some Denisovan DNA.
Right. I know in one of your lectures you described how that happened, and again, it was looking at mitochondrial DNA, where there was one kind of DNA, and then looking at nuclear DNA, and there was another kind of DNA present. So you could see that there was both Denisovan and Neanderthal DNA in the same individual. Is that right?
Yeah, that was a fascinating story. So it was a hybrid. So the fossil remains were of a female, and she had inherited mitochondrial DNA from the Neanderthal. Uh, and as I recall, the to be honest, I don't remember if it was a hybrid between Neanderthal and modern human or Neanderthal and Denisovan, but it was basically the first generation. The
Hmm.
mitochondria was Neanderthal, and the
Hmm.
nuclear genome one was Neanderthal and one was, and again, I'm sorry, I don't remember, either Denisovan or human.
Hmm.
But that was a very striking find and observation that they
Right.
could basically see the first generation of interbreeding between these
Right.
different archaic species.
Well, we'll get it right on. I will dedicate a whole page on my website to this discussion, so we'll clarify that.
Yeah,
I think from my
I know
reading, it was
it's in my book, but I just don't happen to
yeah,
remember what the not the non Neanderthal contribution, whether that's modern human or Denisovan.
yeah. Anyway, we can certainly clarify that because it's just absolutely fascinating. One of the last questions I wanted to answer you or ask you ask you has to do with the use of artificial intelligence and the marriage of that with with PCR technology. What's the future? What's going on with that? I mean, I hear there's been a marriage between the two.
Well, I'm not sure about so much with PCR, but with DNA sequencing information, with next generation DNA sequencing, it's possible to generate huge amounts of sequence data. And the data is so dense and rich that the human mind, the naked human mind, as it were, would have trouble interpreting it all. But with AI, presumably we'll be able to discern patterns and see correlations and make inferences that we weren't able to make prior to the development and implementation of AI.
Mm, understand. And so, corollary to that is what about synthetic biology? I know a colleague of mine from my old college days. I don't know if you knew Craig Ventnor, but
Oh,
he
sure. Yeah.
he has an institute at UC San Diego right now that's I think it's still he's still there working on synthetic biology. And and in synthetic biology, of course, they're trying to create artificial genes and um, even leading to some forms of artificial life in the test tube. Do you have any thoughts on the ethics behind that? I know there's been some controversy about synthetic biology and the use of technology dealing with even PCR technology use in synthetic biology. What do you think about that?
Well, to be honest, I'm not really that familiar with synthetic biology.
Hmm.
I mean, I am familiar with this isn't so much synthetic biology, but it attempts to recreate, extinct species,
Right. Hmm.
by manipulating, say, the genome of a modern elephant so that it more resembled a woolly mammoth or something.
Right.
And of course, PCR would just be a small part of the technology that's involved in that, or as you say, the synthetic biology. Um, I mean, sometimes I think we're creating solutions that are in search of a problem.
Right.
Uh, that
Understand.
so I guess I'm a little skeptical about some of these things, but I guess if they really meet a need, um, I think that's okay. That would be a
Understand.
real contribution.
Well, my last question is what's the future, do you think, of molecular biology? Where are we headed? We had this tremendous tool that you and your colleagues discovered and developed, the PCR technology. What's on the horizon? Is there something going on in the secret labs or in the bowels of laboratories that's going to produce something as powerful? We just saw CRISPR in the last, well, 5 years
Yeah.
actually, come to the fore. And what's out there from a scientist's point of view, from your point of view?
Well, as you point out, Don, CRISPR is certainly a major contribution and a revolutionary technology that makes gene editing and gene therapy more possible. I think for me, maybe as a geneticist, all the new technologies for generating DNA sequencing, there are now technologies that can sequence very long stretches of DNA very rapidly. So there's a technology known as Oxford Nanopore, that takes, maybe an hour or two to generate a sequence that used to take many days.
Hmm.
And so I think we're going to be generating a lot more information, and the technology is not just in carrying out the sequence, but it's preparing the DNA to be sequenced. So I don't know if you come across this article, came out, I think, within the last year or so, but scientists from, I think it's Finland, um, was sequencing genomes that were 2 million years old in a sediment in Greenland.
Hmm.
And so it was inconceivable that DNA that old could ever be recovered and analyzed.
Right.
But techniques for preparing DNA for sequencing from very tiny fragments of DNA have been developed. So to me, that's some of the most remarkable new technology. It's the ability to sequence truly ancient DNA, not
Right.
50,000, not 100,000, but these are literally millions of years old. That,
Right.
I think, well, a few years ago, actually, a few minutes ago, I would have thought totally impossible.
No, I understand. Well, we actually did a show on this. I think I mentioned to you, Dr. Greenwald at the Smithsonian discovered a mosquito, an ancient mosquito,
Mhm.
in a sediment that actually had hemoglobin
Yeah,
in
no,
its
that's
gut.
fascinating.
It was just absolutely fascinating. And now they're actually able to tell a lot about the morphology of dinosaurs and ancient creatures just from being able to sequence DNA like you're talking about. They can see what the proteins were like that would have been produced from that DNA, and then they're able to infer
Hmm.
from that even the color of uh, of, of feathers and the structure of these dinosaurs, and it's just absolutely fascinating work. So, last question: what's the future for Dr. Erlich? Where are you going now? I mean, you've done such wonderful work in your career. What's next for you?
Well, I'm emeritus now, so I still have an office, and I go in and uh, consult with colleagues. That our existing project is actually once again, 40 years later, prenatal diagnosis for sickle cell anemia and beta thalassemia and also alpha thalassemia, but it's based on fetal DNA in the mother's blood.
Right.
So, before when we did prenatal testing, it was amniocentesis or chorionic villus biopsy, which
Right.
confers
Mm-hmm.
some risk to the fetus. But if you
Right.
analyze the mother's blood, of course, there's no risk to the fetus.
Mm-hmm.
So that's one thing. And I'm doing a lot of reading and writing, not just about science, but about politics.
Mm-hmm. Right.
Um, one of the, we've talked a lot about science. I mean, that was the main focus of this discussion. But one of the things I'm concerned about uh, are kind of the attacks on science and expertise in the
Mm-hmm.
current political environment.
Right.
So, we may end up with a head of Health and Human Services who's basically a vaccine denier.
Right.
And the president-elect thinks that climate change is a hoax.
Right.
Um, so these are all things that I'm worried about, the attack on science and
Right.
expertise, really.
Well, so am I, and that's the whole purpose of this podcast, A Journey Through Nature and Science. I just actually rebranded our podcast. I've been doing this for 5 years and focusing on human interest stories with respect to nature and science. But now my focus is more on what you're talking about, trying to get the right kind of science information out there. And of course, I did seminars for years
Mhm.
talking about trying to get people to understand science and get disabuse them of some of the conspiracy theories surrounding science. And now science is under more threat as well. But in some ways, I'm seeing a silver lining in the sense that people like myself, yourself, and others now are going to be a lot more aggressive in communicating uh, the facts, the truth about science, letting people know about all of the positive things that are happening out there as a result of science, whether you're talking about space exploration or you're talking about the inner world of molecular biology, things that you can't see producing tremendous benefits for society. So this has been great, and I want you to keep up that work. And there has to be a positive, I think, communication to people, not just a negative resistance, but a positive and
Yeah,
an upbeat communication to people about what science is doing and how important it is to society.
yeah. No, no, I agree. So keep up the good work, Don.
Well, thank you very much, Dr. Erlich. It was a pleasure to have you on the program.
Well, thanks a lot.
Thank you for joining us on this episode of A Journey Through Nature and Science. We've been chatting with Dr. Henry Erlich. Please don't forget to hit the like button to help us grow the podcast and help spread science literacy. Dr. Craig Wheeler will join us on our next episode to share a provocative message that taps into the core of the human experience, existential, thrilling, and frightening all at once. His latest new book is The Path to Singularity: How Technology Will Challenge the Future of Humanity, which addresses some of the most critical questions we face as a species. Neil deGrasse Tyson, who wrote the foreword to the book, said it best: "You'll never see the world the same way, and that's a good thing." As always, stay safe, share happiness, and remember, never stop questioning. Curiosity has its own reason for existence.